Hi everyone, my name is Trent McCracken. Today I'm going to show you how I use flow cytometry to test chromosome doubling in holly plants, and this can help us identify tetraploids that have unique ornamental traits. Just a quick definition here, a diploid plant has two sets of chromosomes, and this is what most plants have naturally, and a tetraploid has four sets, and this is what can lead to interesting changes in size, shape, or growth habit. Here are the materials that I'm going to be using to prepare the leaf sample. You're going to need the corresponding pipette tips for the ones that were just shown. This is the nuclei buffer. I'm using the cysteine UV Ox Protect from Sysmex, and this is both the buffer and DNA stain. I'll be running samples on this flow cytometer using the CytExpert software, and this is available at the Cytometry Shared Resource Lab at UGA. Here's an example of a piece of young, actively growing holly leaf, and this gives the best results because the cells are dividing more actively and they tend to be more intact. So you'll need this and a standard plant for analyzing data later. We're using tomato because the genome size is well known and the peaks don't overlap with the holly. So I'm putting the tomato leaf following with the holly leaf into the petri dish and adding 500 microliters of the nuclei buffer, and then aggressively chop for 30 seconds to break open the plant cells and release the nuclei. Once the chopping is finished, we're going to let it stand for a minute to release all of the nuclei, and then filter the mixture into the test tube using the filter. And this is what it looks like. Following this, we're going to put a milliliter of fresh buffer directly into the tube to stain the DNA, and when it's run through the machine, it's going to fluoresce or glow under the laser. Now I'm going to run the samples. I'm going to open the CytExpert program. I'm going to select New Experiment from Template. I'm going to browse new experiment and save it under my folder, then browse the templates and select one I've already created for this project. And then I'm going to select OK, and what's going to pop up is the program, and you can see on the left and the right the boxes where the data is going to populate. Once I'm ready, I'm going to insert the tube into the arm, hit record. The arm is going to pull the sample up and it's going to begin fluorescing the sample. And what's being shown here is on the left a histogram and on the right is a dot plot. And the dot plot is showing individual events that the machine is picking up and then making it easier to see visually with the histogram or all of those peaks on the left. Now we're going to interpret the output, which is the histogram. And here are the key peaks when you're looking at this. So the G1 peak is going to be the largest, and the G2 peak is going to be those smaller ones there. So what's highlighted in pink and green are from the diploid holly, and what's not highlighted but just as large next to them are from the tomato. The G1 is where the nuclei are just going about their normal business with a regular amount of DNA, and the G2 is further to the right, and they have copied their DNA and are ready to divide, so they have double the DNA. So in this diploid, the G1 is more to the left. If it were successfully doubled, you would see a larger peak where that green 4x is, and it would be shifted more to the right. Now we're looking at a histogram from a different holly species that we confirmed as a tetraploid. So like before, we're looking at the G1 peak, which is highlighted in pink, which shows how much DNA in the nucleus is at rest. So in the diploids, that was shifted further to the left, but in this case, it is shifted much further to the right, which tells us that the plant has twice the DNA or four sets of chromosomes. And this is the shift that we would expect when a plant has undergone chromosome doubling.